so with that, I'm going to introduce here Peter Krug, who works at the Vaccine Research Center at NIAID. And he's going to tell us about his work on virus-like particle vaccine candidates for enterovirus D68. So go ahead, Peter, take it away.
Thank you, Matt, for that introduction. So before I talk about our virus-like particle vaccine platform, I want to give a little bit of a brief introduction of some examples of the current coronavirus vaccines, because they play a role in how we compare what the products we're making now with the existing coronavirus vaccines. So everybody here is very familiar with the inactivated poliovirus vaccine. This is an amazing vaccine in that it protects greater than 99% against paralytic disease after 3 doses. And as such, it's been used worldwide, 10 billion doses since the turn of the 21st century. Also recently, a new enterovirus A71 vaccine has been licensed in China. This is a virus that causes hand, foot, and mouth disease in children, and it can lead to encephalitis and meningitis. So it's a serious problem there. And this vaccine has been shown to have at least 95% efficacy against hand, foot, and mouth disease in China. And I also want to compare to our counterparts on the agricultural side. There's this vaccine for foot and mouth disease virus. We've learned a lot about how cross protection against the various serotypes of FMDV and how the vaccines have to be modified in order to confer optimal protection to prevent disease. The goal of all of these vaccines is to elicit high levels of potent neutralizing antibody, and that's the correlative protection for these vaccines. So how are these inactivated virus vaccines made? Thanks to Bill Jackson's talk, he basically walked us through this process. And basically, we're going to focus here on this back end of as the viruses are leaving the cells. You get the cleavage of the viral polyprotein shown here that's expressed from the viral RNA. The individual capsid proteins associated into protomers, which assemble into pentamers. These pentamers then assemble into a provirion, which in the presence of a progeny viral RNA, it could become a mature virion after a genome-induced virion maturation process that Dr. Jackson talked about. So the inactivated virus vaccines are made from literally 1,000-liter scale cell culture processes and then chemical treatment to inactivate the virus. The virus-like particle vaccines that I'm going to be talking about are very similar to these immature provirions. And the way we make those is by expressing small parts of the virus instead of the intact viral genome. We transfect cells with plasmids that express just the viral capsid polyprotein, this P1 region. And we co-transfect in the plasmids that express the 3-CD protease. So in transfected cells, the 3-CD protease will cleave up this capsid polyprotein, and we'll get those individual capsid proteins made as monomers. They will associate as protomers, they associate as pentamers, but because there's no infection, there's no viral RNA, and we're stuck with this provirion as our virus-like particle. So the big question then is, will these VLP vaccines elicit similar protection compared to these inactivated virus vaccines, which are the gold standard for coronavirus vaccination? They're slightly different. The virus-like particles are a little bit bigger. They haven't undergone that final maturation process, but they're safer because there's no viral genome, there's no replication. There's no need to adapt an infectious virus to cell culture so you can grow up these large quantities of virus. And you can easily swap out these capsid genes. So in the case of a new virus that may appear in the population, you can have a virus that's matched, or you can have a vaccine that's matched to that virus. And this all feeds into our Vaccine Research Center pandemic preparedness plan. We want to leverage a virus-like particle platform so we could rapidly and flexibly respond to EV D68 outbreaks. So to test the hypothesis, answer the question whether or not these particles are similar, we made large quantities of virus-like particles and inactivated virus. These are all based on 2014 virus outbreaks. The VLPs are based on a Colorado outbreak virus, and the inactivated virus particles are based on a Missouri outbreak virus. After purifying these particles, we show that we see all the bands that we expect in protein gels. These products equally bind commercial EV D68 antibodies, and they equally bind the monoclonal antibodies that Matt talked about earlier that were from human patients. So these are the two products here. On the left are the VLP, and on the right is the inactivated virus. How well do these particles elicit neutralizing antibody against the virus? To answer that question, we set up a prime boost regimen for vaccination of mice. These were vaccinated intramuscularly with different concentrations of either inactivated virus or the virus-like particles. 2 weeks post-prime and 2 weeks post-boost, we took serum samples, and then 8 weeks post-boost, we took terminal bleeds. All of these products were adjuvanted with the Sigma Adjuvant System adjuvant, or SAS. Looking at the 2 weeks post-boost serum, we found that the two highest doses of the virus-like particles were able to elicit similar neutralizing antibody titers in mice as compared to the inactivated virus. But we wanted to, so this told us that at least initially we're seeing similar levels of reactive antibody elicited from these two different vaccines. We wanted to look at this in vivo. So we took the purified IgG from the terminal bleeds and did a passive transfer study comparing the IgG pooled from the inactivated virus vaccinated mice and the VLP vaccinated mice. These passive transfer studies are set up where we transfer different concentrations of antibodies into mice that are susceptible to a mouse-adapted EV D68 that we received from Bart Tarbit's lab at Utah State University. The virus grows to high titer in the lungs and can be found to disseminate to the spleen via the blood. We used the 3 different concentrations of the IgG from the VLP vaccinated mice and 3 different concentrations from the inactivated virus mice. We used Matt's 228 antibody as a positive control for neutralization, and we used naive IgG as a negative control. We take samples to confirm the mice have anti-EV D68 antibodies. You can see that over here. And then later that next day, we challenge with the B1 subclade EV D68 mouse-adapted virus. 2 days later, we take lung, spleen, and serum to look for the presence of virus. What we found in this study was that the 228 antibody completely abrogated virus replication in the lung as we expected. The negative control IgG allowed 6 to 7 logs of virus to replicate. And in our test system, when looking at the in our test samples, looking at the IgG from the VLP and inactivated virus vaccinated mice, we see a nice dose-dependent decrease in the amount of virus replication in the lung. And we see complete abrogation at 30 milligrams per kilogram in this study. We also saw very nice inhibition of dissemination with these two treatments, indicating, again, that the inactivated virus vaccine and the VLP vaccine are eliciting similar functional in vivo neutralizing antibody. But as Dr. Hodgecroft at the beginning of the session described, the B1 subclade viruses from 2014 that our vaccines are based on are no longer circulating in the population. The B3 subclade viruses have replaced the B1 subclade viruses in the B clade. And there's also this A subclade virus, that, specifically the A2 subclade viruses that are currently circulating in the population. How well do the B1 immunogens neutralize these B3 and A2 subclade viruses? And as it turns out, as we've seen in the previous talks, that in Dr. Vogt's talk of the B3 or the 228 antibody neutralizing the B3 viruses, we also see about a 4 to 6-fold hit in the neutralization capacity of the serum raised against B1 immunogens to neutralize the B3, 2 different B3 viruses. We also see a 20 to 40-fold decrease in the ability of the B1 elicited sera to neutralize the A2 subclade viruses. Again, we see similarities between the two different immunogens, but again, this is starting to let us know that the B1 subclade may not be the optimal vaccine for future outbreaks. To confirm this in vivo, we did another passive transfer study. This time, we used a B3 mouse-adapted virus that I made in the lab to do the challenge, to look at the ability of the B1 subclade immunogens to neutralize the B3 virus in vivo. What we found was that we had to basically double the maximum dose, or we had to give the maximum dose of antibody from the B1 vaccinated mice in order to completely neutralize the virus. While we still had great blocking of dissemination in this study, we knew that we're starting to see that neutralization capacity hit, and we see it in vivo. Again, similar effects between the two different immunogens. So from this first part of the study, or for the first part of this talk, we've learned that VLPs elicit very strong neutralizing antibodies that are similar to the B1 inactivated virus vaccines. Both of them inhibit in vivo replication of the B1, the matched virus, the matched mouse-adapted virus. But both of these immunogens are eliciting weaker responses against the heterologous subclades. And this is recapitulated in vivo, where the B3 mouse-adapted virus required much higher doses of antibody to block the lung replication of the B3 mouse-adapted virus. So we conclude from these studies that the B1 immunogens did not provide optimal protection against these heterologous subclades. And really the question is, if you look at the B subclade, how long will these B1 immunogens be protective as the virus continues to evolve? The answer is that we don't want to wait to find out. We want to go ahead and evaluate B3 and A2 subclade VLPs. The rest of my talk, I'm going to talk about the work we've done with our B3 subclade-based VLPs. These VLPs are based on a virus that was a 2018 outbreak virus that was associated with AFM. These particles are very stable, so we're looking at EM. Looking at these particles in EM, they maintain their structure on EM after multiple freeze thaws, and we can store these particles at 4 degrees for at least 4 months, and they maintain their structure. Looking at the immunogenicity of these particles, we wanted to test a bunch of different parameters. So first we wanted to not only look at immunogenicity, but we wanted to look at durability of the immunogenicity. So we did a longer study where we took more bleeds from the mice after the boost. Also, we know that the adjuvant that we used for the first study, the Sigma adjuvant system, isn't an adjuvant that could be used in humans. We wanted to see if we wanted to assess whether or not other adjuvants that could be used in humans would be similarly effective at eliciting neutralizing antibody. So we had 5 groups of mice. The first group was adjuvanted with nothing. The second was with SAS, again, as a control for the first study. Then we used alum in the third group. And then we used 2 different concentrations of a new adjuvant relatively new adjuvant called Adjuplex. And we use this adjuvant because it is known that it provides a more balanced Th1, Th2 response. There is some concern that having a Th1, a Th2 skewed response for respiratory infections might cause some problems. So we wanted to make sure to see if the Adjuplex adjuvant would also provide good protection in our cell culture and mouse systems. All right, so right off the bat, what you're looking at here is the neutralization study in cell culture for all of the formulations and all of the time points. So in each group of the formulations are shown here, and inside each group is the 14-day post prime, 14-day post boost, 28-day post boost, 42-day post boost, and 100-day post boost serum samples. And each point is an individual mouse. What you can see here is that there is, of all of the adjuvanted formulations, there was no statistical difference between any of them. But there was good durability in all of the groups over the out to 100 days post boost. Even though this was just this was designed as an adjuvant selection study, we went ahead and pooled the serum from these two Adjuplex groups and concentrated and purified the IgG for a passive transfer study. Again, these susceptible mice were given different concentrations of either 228 or the virus-like particle pool, the IgG from the virus-like particle vaccinated pools. We saw that all of these mice did have EV D68 specific antibodies after the passive transfer, and we challenged them with a B3 mouse-adapted virus that grows to high titer in the lungs and disseminates to the spleen via the blood. What we found was that the 228 was again very good at neutralizing the B3 subclade virus in vivo, blocking lung replication, whereas the naive control allowed 6 to 7 logs of virus replication in the lung. We saw a nice dose-dependent decrease in the amount of virus replicating in the lung using the VLP IgG from the vaccination studies. Looking at dissemination, in all but the lowest dose of the VLP IgG treatments, we saw no dissemination. Now, looking at cross-neutralization of the B3 VLP elicited sera, we saw something different than what we saw before. So what we saw is that, again, we have this strong homologous response against the B3 subclade virus. The neutralization of a B1 subclade virus took a hit, about a 4 to 6-fold hit again, 4 to 6-fold hit. But interestingly, different from what we saw with the B1 immunogens, we saw that the A2 subclade virus was neutralized just as well as the B1 subclade virus was. This was different than what we saw from the previous study, where we saw this strong homologous response against the B1 subclade virus with the B1 immunogens. We saw this 4 to 6-fold hit against the B3 subclade viruses, and then this poor 20 to 40-fold decrease against the A2 subclade virus. So it's something, and this is definitely a different pattern that we're seeing with the B3 immunogens. This isn't due to the adjuvant that's used. So the SAS controls here also showing this difference between B1 and B3. So to summarize the second part of the talk, we've made these B3 VLPs. They're very stable, and we get good yields of mammalian cell culture. All of the adjuvant formulations elicited high titer neutralizing antibody, and the durability was good, at least out to 100 days post-boost. I didn't get the time to show, but the 20% Adjuplex formulation elicited that balanced Th1, Th2 response, and the IgG that was concentrated and purified from the vaccinated mice blocked lung replication and dissemination of the mouse-adapted B3 virus in vivo. I just showed you that the B3 VLP vaccination seems to elicit higher levels of neutralizing antibody against the A2 subclade virus than what we saw with the B1 VLP vaccination, and this was not due to the adjuvant. Taken together, overall, these results demonstrate that first, that the EV D68 VLP vaccines are as effective as inactivated virus vaccines, at least in the mouse model. And second, that we've identified that we need to optimize EV D68 vaccines to provide protection against circulating strains. We're seeing a neutralization capacity hit against different subclades. And this can be provided either by using something like the B3 VLPs that seem to be naturally cross-protective, or a multivalent vaccine where you mix different VLPs from different subclades together, or even a mosaic VLP that would express epitopes from different subclade viruses. All right, with that, I need to thank the folks in our respiratory virus core in the molecular immunoengineering section. This is led by Tracy Ruckworth. I need to thank Dan Moss specifically for helping me out with the B3 subclade passive transfer study and doing that the TH1 TH2 study that I didn't get to show you. And then I also need to thank the folks in our animal facility that helped us with all the bleeds of all the mice, and need to thank Matt Vogt when he was in James Crow's lab for the antibodies that they gave us for our positive controls. And a shout out to NextStrain for all of the great study, or all the great phylogenetic analysis of these different viruses, so we can try to figure out why the B3 VLPs are working better at cross protection than what the B1 VLPs are. And with that, I'm happy to take any questions. Thank you.
Thanks, Peter. That's really encouraging data, certainly for me on many fronts, because, I think obviously the idea of preventing AFM before it ever happens is the most, the thing that we would all like the best. But also, seeing some of that cross-reactivity from 228 certainly warms my personal heart. So thank you for showing that. And then thank you for also bringing us all the way back around in a full circle and having NextStrain be the final thing that you cite after having Emma Hudcroft give the first talk, the person who is in charge of NextStrain for D68. So super exciting session. All right, so let's get to some questions. First question for you, Peter. In addition to the inactivated and VLP vaccine candidates discussed, is there any interest in developing mRNA vaccine candidates? And if so, have antigenic targets been identified? So is there any hope for a pan-EV vaccine using a common epitope shared between, all the various enteroviruses?
So that's a very interesting question. I'm sure that we are not doing any mRNA studies right now. I'm sure there are a lot of companies out there that are probably very interested in doing that. One issue is that you would basically need to express the entire capsid, I believe, of the virus in order to generate the proper neutralizing antibody responses. Neutralizing antibody requires that structure of the three-dimensional structure of the capsid. So it's probably going to be an expression of the entire P1 region of the virus, and that would have to be cleaved by a protease.
Yeah, I tend to agree with you on that one, Peter. I think those linear epitopes that Bernardo spoke of are hopefully going to be very helpful from a diagnostic perspective. And I think Bernardo showed some great data to indicate that, but I do question whether or not they would work from a virus or a vaccine perspective, but that would be really cool. All right, I also, let's see. So that last section where you sort of compared, I think it was the last set of data you showed, where you were comparing the B3 immunogenicity and cross-reactivity. This one, where it did better against the A2 than the B1. Are these different adjuvants as well, or are the adjuvants the same and it's only the sequence? I just couldn't remember.
So in here, you can see that the 2 concentrations of Adjuplex are used for these 2 sets of neutralization.
Okay, and then
But this is the control for the previous study. So this was all done with SAS with the B1 VLPs. This is B3 with SAS.
Yeah. So do you think it could be that not once B3 has become dominant, the cross-reactivity that natural infection makes might be better than, you know, the B1 clade when it was circulating? Might that be an indication here?
I think that these are the kinds of questions that Dr. Hodgecroft would be better at answering. I mean, we've done some initial looks using the next strain data, and it does look like there may be some specific amino acids that are similar between B3 and A2 that are not similar between B1 and A2 in the variable regions, like the DE loop and the BC loop. But what we don't know is the effect of any one single amino acid change in those loops and how it would affect the capacity of antibody raised against them to neutralize other subclade viruses. But yes, it's definitely possible.
All right, and since you had to wait till the end, I'll get one more question in and go a little over time. Our fellow panelist, Bernardo Mino, has asked, do you have any plans to make like a multivalent D68 vaccine to try to, you know, put all the clades in or something like that?
Exactly. So that would be probably provide the best protection. So if you knew that there were different subclades all at the same time, and you needed to have that optimal protection against all of them, yes, a multivalent vaccine would be something like the poliovirus inactivated vaccine, where you have 3 different serotypes in that. So yes.
Yeah. Well, thanks. I think this ends on a really high note for me. And actually, if I can have you unshare, because I think Carlos is going to share a slide now. But just, I think that to me is a really positive note for everybody who's listening, hopefully, that, you know, I think this could be a vaccine preventable disease. Your data are fairly compelling, I would say. And I want to use this chance while I have the soapbox for like 1 more minute for especially all the clinicians on the call, especially those people who are the first people to see someone who comes in with a case of AFM. We talked about this a little last week, but I think this gives us the chance to reemphasize a point that was made. Get those patient samples early, as soon as you can, for specimens from their noses, from their GI tracts, from their spinal fluid, from their blood. What I think you've seen in all of the talks today, I'm looking at all the speakers, I think all of us have referred to the fact that we've used this strain or that strain, and some of these are historical strains and some of these are new strains. And, you know, the more samples we have banked, the more powerful our chance to do the research becomes, and gives, you know, those of us who like to play in the laboratory a lot more to work with. And so just to reiterate to everybody, at least in the United States, if you have a patient you suspect of AFM, there are not 1, but 2 different studies that you can get involved with. So if your university or your institution is part of one of the sites of the AFM natural history study that's sponsored by NIAID, that's probably the easiest route in, because that has a pretty strong infrastructure set up to collect those specimens and bank them for future studies. But anybody can get involved. So the CDC is also sponsoring a different sort of biobanking study that if you're at any other place that is not one of those sort of 30 plus places, or maybe it's even 40 that the natural history study has, anybody can get involved. And so I just did this exercise just to prove to myself how to do that. If you just Google CDC AFM research, you'll find links to both of those studies and information on both of those studies. So pretty easy. Google CDC AFM research, collect your samples early, collect your samples often, and we'll have even cooler, newer seminars in the coming years because we'll have such awesome specimens to do more research with. All right, thank you to all of our panelists. I feel elated that there's just so much cool research going on, and I think you all have done a really great job of putting together a common theme of further trying to understand how this disease works. All right, so I'm going to shut up. I'm going to let Carlos take over and talk about what we've got in store for our next session.